Everyone, welcome to the class. Today we will be dealing with the mechanism of enzymes. So if we talk about the mechanism of enzyme, first we will see the enzyme action. Enzyme enhances the rate of the reaction. Suppose this is a reaction, A plus B changes to C plus D, and this reaction in the absence of enzyme, suppose it takes 10 hours, right? But if you add the enzyme, what will happen? The speed of the reaction will increases and the time duration will decrease to 20 minutes. So we can say that what we can say is the enzyme enhances the rate of the reaction without taking part into the reaction, right? Enzyme binds to a given substrate material to produce the desired product. If this compound I am considering as S, and this is the enzyme. So what enzyme will do? Enzyme will form a bond with substrate, resulting in the formation of ES complex, and this ES complex will change to product later on, right? And this, from the product, the enzyme gets separated out and you will be getting an individual product. So we can say that now here you can see the enzyme is, what you can see here, enzyme binds to a given substrate. So here enzyme is binding to a substrate. Why? To produce a desired product, that is P. When the enzyme binds to a substrate, it forms an intermediate complex before the final product. So these are the intermediate complex which is formed after binding of enzyme to the substrate, right? So the reaction takes place as follow. Enzyme reacting with a substrate leads to the formation of enzyme substrate complex, right? So now there is a disintegration of the complex molecules to give the product. So ES will change to EP, individually it will change to enzyme plus product. So this is how you will be getting the product, right? Coming to this is, you can see enzyme reacting with a substrate, which as a result it will form ES, enzyme substrate complex, which later on modified to enzyme product and then changed to enzyme and product, right? So that is what it is, what we call the action of enzyme. How enzyme act? Enzyme, what it will do, suppose A, we want to convert it to C. So in the presence of enzyme, how we will do it? Suppose this is an enzyme added. So what enzyme will do? It will first form bond with A and form a complex EA. Later on it changes to EC, and after that you will be getting a C product. So we can say now A got converted to C, right? So this is how the enzyme works on it. Clear everyone? How enzyme work? So we basically, this part, the substances which are present for the reaction, which initiate the reaction, we call it substrate, and that's why we are using it S. And this part we call it product, and for product we are using P. Clear everyone? Yes, that is how the reaction takes place. Coming to the mechanism, how the enzyme decreases the your time duration or increases the speed of the reaction. This is because if you see here, this is energy and this is reaction coordinate. Here it is called reactant, which we also call it substrate, and here it is product, right? If you do not add the enzyme, see here, without enzyme you can see the energy is, if you see the without consumption, the energy level, it is very high. Without energy, you can see this is the action of energy level without enzyme. And if you see, this is something with enzyme, this is something with enzyme, and if you see the with enzyme part, the energy consumption is less, right? So if you can see here everyone, this is overall energy released during reaction. This is the activation energy without enzyme, and this is activation energy with enzyme. So what you can see is the overall energy is remaining same in both the cases, but the difference is only with the activation energy. In one case the activation energy is high and the other case the activation energy is low. Now the question is, what is activation energy? What is activation energy? If we define activation energy, So I think you have studied about activation energy in class 11th and 12th also, right? So activation energy is basically what? It is the energy, it is the maximum, we can define activation energy as the energy required by a reacting molecule to get converted into product. Activation energy is what? Energy required by a reacting molecule to get converted into product. Energy required by a reacting molecule to get converted into the product, to get converted into the product, right? Or you can also divide activation energy as minimum amount of energy needed to activate molecules so that they can undergo a chemical reaction, right? So this reactant to get converted into product, they need certain amount of energy, right? And those energy, the amount of energy, those amount of energy which is needed by the reactant to get converted into product, we call it activation energy, right? If there is no enzyme, in case of without enzyme, you can see the activation energy is large, it is required in large amount, right? But if you see with enzyme, when enzyme is added, if you see the activation energy is less. So we can say that enzymes, what we can say is enzyme reduces the activation energy. Enzyme reduces the activation energy, right? So we can conclude from this graph that enzyme actually reduces the activation energy and promotes the fast conversion of reactant into the product, right? Okay, this is the mechanism of enzyme action. Enzyme increases the reaction rates by decreasing the activation energy. What is activation energy? It is the energy required by the reactant molecules to get converted into the product. So the formation, say whenever enzyme is reacting with a substrate, it will form a bond with substrate and form a complex ES, right? This is later change on EP, which now again change to your enzyme plus product. This is what we have seen. Now this enzyme substrate complex, right? This enzyme substrate complex, formation of this complex is described by two theories, that is lock and key model and induced fit model. We will see one by one, right? Okay, so lock and key model was given by Fischer. Fischer was a scientist who gave the lock and key theory of enzyme, and induced fit theory was given by Koshland, right? So these are the two scientists who gave the theories for enzyme action, how the enzyme will bind to the substrate. According to lock and key, what will happen, like every lock, if this is a lock, this lock will be having a key, right? If this is a lock, you can say if this is a lock, this lock will be having a key, right? This is a lock, this lock will be having a key. If this is another lock, right, this will also be having a key, right? So every specific lock is having a every specific key. Every specific lock is having a specific key. Similarly, every specific enzyme is having a site for the substrate, and that substrate is specific for enzymes, right? That is what actually lock and key theory describes about enzyme substrate complex. If you see, this is an enzyme and this is the active site. Active site means what? This is the site which when binded by the this is the site which when bounded by the substrate gets converted into enzyme substrate complex, right? So if you see, this substrate is getting insight into the enzyme, as a result enzyme substrate complex is formed. Now what will happen, there will be a formation of enzyme and product. After that, product will be released and enzyme will also be released as a separate entity, right? This is what is your lock and key. But if you say induced fit theory describes that the active site of enzyme keeps on changing. It states what active site of enzyme keeps on changing. You can see here this is one shape, right? And if you see here this is another shape. Clear? So we can conclude from induced P theory that binding of substrate changes the active site of the enzyme. You can write binding of substrate changes the active site of enzymes. Right? Now coming to lock and key hypothesis. So this is lock and key hypothesis. Already I have explained you in a very simple form. Now if everything is explained here in detail form, we will say this also. So lock and key theory was given by Fisher. Fisher was a scientist who proposed the theory called lock and key to describe the mode of action of enzyme, how the enzyme works. According to this principle, if the right key fits inside the right lock, the lock will be open, otherwise it will not, right? Everyone knows if you have a when you lock your home, when you locked your home, what happened after some time? If you don't get the proper accurate key, you will not be able to open the lock. So what you have to do, you have to call a key maker to open the door, right? So that means what? Every specific lock is having a every specific key. Similarly here also what happened? Every right enzyme fits into a right substrate. That means if this is the enzyme, so the substrate will be specific for this enzyme, right? That is what it actually defines. Enzyme shape provides surface configurations that can fit with the other molecules. The molecules on which enzyme act are known as substrate of enzymes. So this is known as enzyme, right? And this enzyme will be working on, see, suppose if, let me tell you, if this is a reaction A plus B changes to C plus D, right? Now if this is the enzyme E, now this is the reactant and this is the product, right? So enzyme will obviously react with the reactant. Why? To get the desired product. Now the you can say the molecules, the molecules on which enzyme act, means here in this case the enzyme is going to act on this molecule. So this molecule we call it substrate, right? The molecules on which the enzyme act is known as substrate of enzyme, right? So these are the molecules here. Here the enzyme will act and gets and here the enzyme will act. So we call this molecule as substrate. And the substrate is specific for its enzyme according to the lock and key hypothesis. I hope it is clear. Next, substrates that have the correct geometric shape can fit within the active site of the enzyme. The active site of the enzyme is highly specific about the surfaces of its substrate. So if this is an enzyme, say, if this is the active site, suppose this is the active site. Now what will happen? This is very specific. This active site is very specific for substrate. It can fit only with those molecules which is having a shape that can fixed into it. If like this type of structure will never worked on this enzyme, right? So that's what was explained by the Fisher. The enzyme and substrate fit like a lock and key, making it a lock and key enzyme action model. Sometimes, however, certain molecules close to the substrate may also interact with an enzyme active site. The molecules competes with the substrate, and now sometimes what happen if this is an enzyme. Now for substrate I am using this blue color, and for inhibitor I am using this color. Now what is happening here? Say, okay fine, let me change the shape a little bit. Okay fine. So this is what we call substrate and this is something we call inhibitor. That means which will inhibit the enzyme, and this is what we call enzyme. If enzyme forms bond with substrate, then it leads to the formation of enzyme substrate complex, which will later on form enzyme product, right? But if this is inhibitor, this also if this also will be having a tendency to bind with this, right, active site. So if it bind to this, then there will be no product. Why? Because this is inhibitor, right? both of them is having only one active site for binding. So what will happen? The molecule competes. You can see this part, the molecule competes with the substrate. So these molecules will now compete with the substrate and can either slow down or stop the reaction. So if it is if the inhibitor gets bound to the enzyme, then it will stop the reaction, and such substances is called competitive inhibitor. And because this inhibitor is competing with substrate to bind to this active site, right? So we call this mechanism as a competitive inhibitor. As what? As a competitive inhibitor, right? Because it acts to prevent the product from being created. Why we call it inhibitor? Because there will be no product formation. Clear, everyone? Yes. So let me tell you once again here. If you see this, if this is an enzyme, right? This is an enzyme. Now this is, say, this is the substrate and this is product. This is substrate and this is your inhibitor, right? You can see both of them can bind to only one site of enzyme. Which side? That is, both of them can bind to only one site, that is the active site, right? This part we call it active site of enzyme. Now what will happen? Since there is only one site present at the enzyme, so what will happen? Both the substrate, both, you can say, both this just a minute, give me a color. Both this substrate and inhibitor compete with each other. Why they compete with each other? To bind to the active site. To bind to the active site. Once they bind to the active site, if substrate binds to the active site, it leads to the formation. It leads to the formation of what? Enzyme product, which will later on change to your enzyme plus product. But if in case inhibitor binds to the product, if in case inhibitor binds to the enzyme, what will happen? It will change to, there will be no product. Let me write here, there will be no product. That is what is happening, right, in lock and key. So this is key and this is lock. So this is substrate and this is active site. This is an enzyme. Once it binds this substrate, once bind to this part, it leads to the formation of enzyme substrate complex. And this is lock key complex, right? Coming to induced with hypothesis. Images, Google. Source, Google, right? Okay, induced with hypothesis. According to induced with hypothesis, the enzyme changes shape. The enzyme changes shape to enhance the ability of the enzyme to catalyze the chemical reaction. That means what? Whenever the enzyme is active, whenever say this is an enzyme, right? So this is, you can see, this is shape and see this shape, right? See this part and see this part. So what we can see that the shape of the enzyme is getting changed, right? Now the question is why the shape of the enzyme gets changed. The shape of the enzyme gets changed to enhance the ability of the enzyme to catalyze the chemical reaction so that it can bind to the substrate and catalyze a chemical reaction. And this induced feed is the most accepted theory for enzyme action. Most accepted theory for enzyme action, right? That's what it's explained in this with hypothesis. The enzyme returns back to its original shape after the reaction occurs. One substrate will bind with this enzyme. Enzyme will change the shape, forming enzyme substrate complex. After that, what happened? The enzyme will be released and this substrate will again come back to its original shape, right? That is what is explained in the induced feed theory, and this theory was given by Koshland, by Koshland, right? That is what induced feed model. If you see here, this is a substrate and this is active site, right? Clear, everyone? If you see this is substrate, this is active site. Substrate enters active site of enzyme and it results in the formation of enzyme substrate complex. Now substrate is converted to product. Now you can see these are the product. Once the products get released, the active site will be remain free. So enzyme change shape slightly as substrate binds. Clear? Yes, everyone. So that is what happened. You can see the shape. You can see the shape, right? It is getting changed. So that is what is explained in induced fit model. If we go for a quick summary, we have seen that enzymes are proteinaceous in nature. Enzymes are proteinaceous in nature, right? So if we move on to the enzyme action, if we see the enzyme actions, what we can see in enzyme actions, we can see that what? We can see whenever there is an enzyme present, it will bind, say let me give an example of a reaction here. Suppose this is a reaction A plus B changes to C plus D, and this part I am calling it substrate S, and this part is your product, which is defined by P. Now what will happen in the, this is an enzyme. So how this enzyme will carry out the action is like this enzyme will form bond with substrate. This is enzyme. Now what it will do? It will form bond with substrate, resulting in the formation of enzyme substrate complex, right? And this enzyme substrate complex gets converted into enzyme product complex. After enzyme product, what will happen? You will get the product and enzyme will be eliminated out, right? This is how the enzyme works, right? Now you can see formation of bond, formation of bone releases energy. Let me write it clearly. Formation of bone. What the formation of bone do? Bone does, it releases energy, right? And formation of bone releases energy, and you can say breaking of bone what? Increases energy. Breaking of bone increases energy. Now you can see this enzyme. If you see this enzyme, this is forming bond with substrate. If it is forming bond with substrate, that means formation of bond what? Energy releases. So I have told you that there will be decrease in activation energy. So how enzyme causes decrease in activation energy? Because once the enzyme forms a bond with substrate, the amount of energy will be released out, right, resulting in the formation of ES complex, and this ES complex gets converted to product and enzyme, right? And what is if we draw the graph here, just a minute, if we draw this graph here, right, this is we call energy, and this is the reaction molecules, and this is say reactant, and this is say product. Now what will happen if this is reactant and this is product? This is normal case. Normal case means what? That is without energy. This is normal, I mean this is, we can write without enzyme. This is without enzyme, right? Now if I add an enzyme here, what will happen? Whenever I add an enzyme here, you will see it gets reduced to this. Now this part is what? This part is basically in the presence of enzyme, right? So you can see the difference here. In the presence of, if I draw it here, in the presence of enzyme, the energy consumption is less compared to the absence of enzyme. In the absence of enzyme, you can see energy is higher, and in the presence of enzyme, energy is less. How it is less? Because it forms bond with substrate, which will release the excess energy. So we can say that enzymes reduces the activation energy. Activation energy, and this is what we call activation energies. Activation energy, right? It is higher for without enzyme and lesser for the with enzyme case. And what is activation energy? If somebody asks you, how will you define? Yes, this is activation energy is basically the energy required by the reactant, to get converted into the product. To get converted into the product, right? That is what is our activation energy. Next, we have seen two theories given. That is, what are the two theories? Yes, two theories we have seen is your lock and key theory and induced fit theory, right? If I write here lock and key, and if I write here induced fit theory, how it works is, see here everyone, lock and key theory was given by Fisher, and this induced fit was given by Koshland, right? Lock and key means what? Every enzyme is having a specific substrate. Every enzyme is having a specific substrate, like every lock with a specific key. But if you say this induced fit, enzyme changes its shape after binding with the substrate, right? That is what is explained by the two theories, right? Okay, so I am ending up the class here. Next class, if possible, we will be dealing with the kinetics of enzyme. Thank you so much, students, for watching this session. Thank you.